Hello, my name is Omid Karkouti. I'm the co-founder and chief operating officer at Rarebase, a public benefit biotechnology company based in the San Francisco Bay Area. At Rarebase, we set out to identify treatments and cures for disorders that fall beneath the typical threshold of biopharmaceutical investment. And we do so by partnering with patient organizations like KIP1A.org that are sponsoring early stage research. My co-founder, Ono Faber, is a rare disease patient, and I'm a rare disease patient advocate. And so, in a way, we are consumers of our own company and program. And we're excited to participate in the conference today and hope to shed some light on our ideas, progress, and plans for the development of treatments for KIP1A associated neurological disorder. Since we're at a patient organization-led conference, I'll comment that we're very excited as a company about the emergence of patient organizations as research hubs, not only providing community support and family and patient engagement and awareness and fundraising, but really extending into building the network of subject matter expertise and clinicians and connecting researchers and research findings across the public sector and the private sector in biotechnology and pharma. It's really impressive, and it's part of the reason that we started Rarebase in the first place, knowing that patient organizations have really taken a driver's seat position for the early stage funding of early stage research towards discovery of new therapies and sharing those findings with the research committee. So excited to be doing that here today. Now, that said, the R&D landscape is pretty complicated. Going from an early stage discovery all the way to a clinical trial is a long process. And Ono and I found, both when advocating for for the discovery of treatments in our own individual disorders, that there was quite a few inefficiencies and misalignment of incentives in early stage discovery research that made it go slower than it really needed to be. And in particular, in on the left-hand side of this slide, you see the space between therapeutic discovery and preclinical efficacy. This is the high-risk area of identifying potential treatments, many of which must be tested in different types of research models. And there needs to be tests that are validated that establish whether a given intervention or a treatment is effective. This could be in cells, could be in animal models of the disorder. There's also a series of biospecimens and unbiased data that may be collected from those things. And each one of these are kind of hard to independently produce. And in many cases, if one institution works with another to generate these things, it's hard for them to collaborate. So when we first started out, we were focusing on some of the infrastructure efficiencies and applying biopharmaceutical best practices towards the development of these resources so they were readily shareable and available to others in the research community. And beyond that, we started to dig deep with our team of scientists and engineers into individual disorders, and even early on started to see patterns between disorders. So since we first started, Rarebase has looked at about 115 disorders in the context of families and patient organizations that are advocating for their patients in the community. So what we really found from that initial survey is that about 2/3 of the connections that we were making were in central nervous system disorders, and that's a primary focus of our company. So a lot of the tools, the methods, the assays that we're developing now are really fit for purpose to address disorders of neurodevelopment and neurodegeneration, of which really fits well with KAND. But I will comment also on, in red on the right-hand side, that patient organizations have really gotten involved with clinical research as well, not only as participants or subjects of research in clinical trials, but also the organization's involvement in the natural history study funding and organization and enrollment and engagement, retrospective clinical research on medical records to try to establish that natural history, and then also being involved in the discussions of the decisions around identification of clinical endpoints and biomarkers. And we get involved in some of this, but most of our focus as a company in this stage is in the discovery phase. So the future state and what we intend to build together with our biotech, with our foundation partners, is to see the next level of patient organization involvement, and as really seeds for biotechnology innovation. And so, with the infrastructure that I was describing before, we see the combination of patient organizations and the infrastructure that we're building being able to really direct and be very efficient with the interest and the funding that's applied towards research. So, as an example, because of long-standing efforts and relationships that myself and others in at Rarebase have really built in support of pharmaceutical and biotech drug discovery programs related to outsourcing, we have access to hundreds of laboratories that are available to do very specialized and high value and high leverage work in support of drug discovery. And to help move that forward, our company's employed and engage a in-house team of creative scientists and engineers that are coming up with innovative ways of applying both technology to biotechnology to try to discover new potential treatments. We also have a number of partnerships with biotech, both pharmaceutical as well as technology, that give us access to really, really cutting edge methods and research tools that can be brought to bear in rare disease when they otherwise would not be. And then because we sit in the middle and we've looked at so many different disorders, we see patterns and identify potential synergy. So there may be a single molecule or compound or drug that might work across multiple disorders, and we have a chance to see that because we're paying attention to very, very small patient populations where others might overlook them. In addition, some of that infrastructure about creating and distributing research materials is really just to make things go faster. So we're very open with sharing. And then as a company, we one of the core tenets was to have very fair and open contracts that support a return on sponsorship investment in research in the case of commercialization, and models around some of the industry leaders in venture philanthropy. And I think that this has a lot to do with the excitement around collaborating with KIP1A.org on the potential for therapies in KAND, applying a number of these different things, these different elements, and trying to give more leverage and running programs more like a virtual biotech. So when we think about KAND, it's a spectrum disorder, of course, and the spectrum of symptoms, encephalopathy, neurodegeneration, epilepsy, and autism. This is important to pay attention to because there may be different therapies that address different elements of this, and also the spectrum of genetics being both autosomal dominant and autosomal recessive, and having a wide range of a wide allelic spectrum of a phenotype or clinical presentation, different variants and different severities is important to pay attention to, because when we're thinking about a therapeutic roadmap, these details call for multiple different approaches to try to encompass the different areas of disease and disease progression. So on this slide, I'm showing an example of a high-level therapeutic discovery roadmap that we've generated, and these are living and they change and as we learn things. But at the moment, there's really three different aspects to our interest and investigation in potential therapies for KAND. And on the left-hand side, there are a couple of descriptions of novel potential therapies, the projects that we have initiated, either partnered with KIP1A.org or independently done some work on our own, or are shaded in blue. And the rest are some ideas that we'll present now, and hope that we are generating additional shots on goal for the identification of treatments. On the right-hand side is a platform that that we're developing at Rarebase to address neurodevelopment and neurodegenerative disorders by applying a series of technologies, by applying both technology, engineering technology, as well as laboratory automation and stem cell science and next generation sequencing to identify potential treatments as well. And we'll go into that a little bit too. I wanted to call out a really interesting paper from Camp Lab in Neuron in 2019, where a series of genes in neurons were knocked down using a modified CRISPR method called CRISPR-I, or CRISPR interference. And this is great. This is a great data set because we happened to be reading this paper and saw KIP1A in there, already knew KIP1A.org. So this is a really, really interesting thing to report on. What this really expresses is the phenotype that KIP1A deficient, both iPSCs induced pluripotent stem cells, as well as KIP1A deficient neurons, have a measurable phenotype in impaired survival, in neurite outgrowth, and also the iPSCs themselves have growth deficits. So this is a really clear phenotype. And when you look across from left to right, there's a series of time and days over the time in cell culture for these different cells. And blue means blue means a decrease, and red means an increase. And you see a decrease in neurite length, you see a decrease in neuronal survival and iPSC growth over time with KIP1A deficiency. So what that really leads us to is the what we've partnered on with KIP1A.org. So last year, our team and scientists identified a target that's relevant to KIP1A associated neurological disorder, neurodegeneration, as well as the hereditary spastic paraplegia phenotype. And we started the development of antisense oligonucleotides against this target. And we shared some of this information with KIP1A.org. And after discussing with them their goals for sponsoring research, we partnered on the development of these potential therapies in March, and really excited to work together. I can say that the work that was done last year was really around the target identification, design, and assays around the target. The current ongoing work is to establish robust efficacy assays for neurodegeneration, and then also the establishment of the KAND patient-derived cell-based models and phenotypes, so that we can really reproduce the something like the data that I showed in the previous slide, and then optimize the target knockdown through our ASOs in preparation for an efficacy study and optimization of nomination of those candidates into a lead, and optimization of a lead in the 3rd and 4th quarter of this year. And we hope to be in planning and intend to move forward, provided the data are good, into further efficacy studies at the end of the year or into the next year in different model systems. But I'm very excited to partner with KIP1A.org on this project, and we'll be providing additional updates as the the meat of the work is really around the establishment of robust quantitative cell-based assays and the establishment of the models and phenotypes that we're going to run the assays in, and nominate a lead from our candidates. In addition, also from the same Campman Lab paper in Neuron, there's a interesting observation on the types of genes that are upregulated when KIF1A is knocked out. And those happen to be mitochondrial cytochrome cytochrome genes, MTCO1, cytochrome oxidase 2, 3. They're upregulated with KIF1A deficient cells, so it suggests a link between the deficiency. And of course, mitochondrial dysfunction is a known mechanism of neurodegeneration in other disorders. And interestingly, there was a report in Journal of Clinical Neuroscience in 2018 that associates mitochondrial dysfunction with a KIF1A mutation in PIHO syndrome and decreased activity of mitochondrial respiration. So this is a really interesting correlation, and we think that this is worth following up. So our intent as a company, because we're already generating some of the research tools, the iPSC neurons from KAND patients, our intent is to look at some assays for cellular energy production, respiration, reactive oxygen species, and see if we can demonstrate a difference between KIP1A deficient cells and controls. And depending on the nature of that dysfunction, we'll select from our library of compounds that we're currently investigating for mitochondrial disorders that we're working on, and see if there might be a chance from that field to rescue the bioenergetic phenotype and perhaps also improve the neuronal survival. So successful compounds from if the assay and the difference between the different types of samples holds up, then any successful compounds that are screened in this manner could be tested in other types of KAND models, for example, in mice, which is we're interested in looking at this more closely because we think there is some potential based on the published data that I presented. Now, I also wanted to introduce you to a screening and computational drug discovery platform that Rarebase is building in response to our more than 2/3 of the disorders we're looking at being involved in neurodevelopment and neurodegeneration. So Function is, as I mentioned before, a CNS drug discovery platform that's integrating methods of stem cell biology, lab automation, next generation sequencing, and computational biology, machine learning, and AI. So it's a lot of different disciplines that are integrated and merged together to try to both predict potentially effective therapies based on wet lab data, experimental data, validated in patient-derived cell-based models, and then learn from that and generate a learning loop. We'll talk a little bit about the predictions because there's some preliminary work that's relevant to KIF1A deficiency that I can present. But if you'd like to learn more about this, this is a program that we are launching, and we'll only cover one aspect of the prediction side in this discussion as it's relevant to KAND. So this one of the screening pipelines that we are running. It's a compound screen in neurons, which reads out in the drug actually is read out in the form of a gene expression signature or transcriptome that's run through a computational pipeline that makes predictions about the potential efficacy of a compound in a given disorder. And because some number of KAND is caused by autosomal dominant variants, the target for this type of screen is the upregulation of gene expression from that good wild type copy in the case of a haploinsufficiency, or perhaps even in the recessive inherited genotypes that may have a partial loss of function. So really looking for upregulation of gene expression so that you get more KIF1A and hopefully address some of the underlying biology that causes the wide spectrum of symptoms in CAND. So I'd like to show you a little bit of preliminary data from a very small compound library that was run in, and we analyzed and looked for compounds or drugs that upregulate KIF1A gene expression. This is really interesting because we've done this for other genes as well, and KIF1A is particularly rich. It's pretty cool. Some genes we don't see anything, some genes we see a couple of things, but this is the one that we've seen the most hits, which is really exciting. Of course, this is preliminary, very preliminary data. It needs to be repeated, reproduced in relevant cell-based disease cellular context, but we're excited about the potential for small molecule upregulation of gene expression of KIF1A. And so our intent is to see if we can reproduce these data in CAND patient-derived iPSC neurons, and then of course also go back and do larger scale screens. So that's really the interest, is that we may have potential drugs or drug targets that might be useful to develop further in particular in haploinsufficient CAND cases, but also in some recessive cases. And there are other elements of the platform that we won't cover today, but happy to discuss. We need to further validate these hits before making them more generally known, and most of them are actually tool compounds. These aren't really representative of approved drugs. But for more information about this, feel free to send us an email. So in summary, we've, as I mentioned, we've partnered with KIF1A.org to develop antisense oligonucleotides that are specifically targeting CAND, neurodegeneration, and hereditary spastic paraplegia. And really excited about the potential for that project, and excited to report back, hopefully at the next meeting or through KIF1A.org's normal newsletter and correspondence as we make progress towards that. In addition, because we've been paying attention to CAND and KIF1A, we're looking at additional avenues to therapeutic discovery, including the mitochondrial dysfunction and also upregulation of KIF1A gene expression as a potential path towards treatment of CAND. And I will also do a little bit of plug about hiring. So we are hiring at Rarebase, and as of this taping in July 2021, we're hiring a head of computational biology and a senior scientist. Please check us out at rarebase.org/careers. If you happen to be in a position to apply, we'd love to have you if you're a scientist or bioinformatician. And if you happen to have some folks in your network that are looking for something, that are looking for a move, we'd love to connect with them. So appreciate you passing this along within your networks. And finally, I want to say a big thank you from our team of patients, patient advocates, scientists, and engineers. We're pushing really hard to move forward treatments for rare and ultra rare disorders, and really appreciate the engagement and the involvement of the patient communities in these efforts. If you have any questions, please don't hesitate to reach out to us. And really, again, thank you all as a community, and the scientists that are supporting the community, and the clinicians on the line that are supporting the community. We're really grateful to have a seat at the table, and hope that we can bring some interesting ideas and technology to bear on KIF1A associated neurological disorder. Thank you.